Hello. So I want to talk with you about the project I worked with at George Dander's lab. Basically, I studied grafting and micrografting protocols of the model species Nicotiana ventamiana, and grafting and micrografting, which is the version, like more specifically working with smaller and younger plants called seedlings, are important tools since they make possible to study the movement and also the phenotypic effect of small molecules in the scion and in the rootstock. In the case of Nicotiana ventamiana, George and his colleagues previously developed through gene editing tools Nicotiana ventamiana line harboring a mutation in the gene A622. A622 codes for an enzyme involved in nicotine biosynthesis, and they found out that this A622 knockout line was not only low nicotine producing, but also smaller compared to the wild type at 22 Celsius degrees. So now we have a hypothesis, and it's that nicotine might be involved in plant growth. Therefore, if we graft an A622 scion into a nicotine producing wild type rootstock, that may rescue the dwarf phenotype of this A622 mutant. The first step to test this hypothesis is through grafting and micrografting trials. For the micrografting, we start using 8-day-old seedlings following a protocol by which nothing but the culture media itself was used to join scions and rootstocks together, but then we switched to a modified protocol by which we use entomological needles to fix scions and rootstocks together in the recovery plates. And also we use an extra pair of needles to go through the cotyledons, like to fix them, so that the chances of scion and rootstock to go apart from each other were smaller. For all the grafting trials, we test grafting success 3 days later using a stereoscope. And on the other hand, we also made grafting trials using 4-week-old plants. We also work with the wild type rootstock, but also with the A622, the PMT, and in this case we also use an A622 PMT double mutant developed through gene editing tools, and we follow this direction, either a wild type rootstock joined to an A622 PMT or a double mutant scion, or vice versa. We use 2 methods, but of these methods we repeated twice. First one was the transverse cut and bud method, plus a grafting clip, like to secure scion and rootstock of all the lines and combinations evaluated. And then we reuse the rootstocks to use a different protocol called wedge and cleft. Around 48 hours later, after the graftings were done, we test graft success. Okay, so when it comes to the results, we evaluated that the micrograftings actually had relatively low success, since not only scions and rootstocks were apart from each other 3 days after the micrografting trials, but they were also going through the end of the cotyledons, also darkening of the cotyledons. We also saw adaptations root formation, which is a common phenotype in these micrografting in Nicotiana ventamiana, according to literature. Okay. So out of all the micrografts done, we only have 1/2 micrograft, which is now growing in soil, that was transferred to soil, and it belongs to the combination of nicotine producing line being the rootstock, that is the wild type rootstock, and a low nicotine producing line, either A622 or either PMT, producing the scion. On the other hand, talking about grafting trials, we also evaluated the success, and it was pretty low, not only using the transverse cut and bud alignment, but using the wedge and cleft alignment, which seems to work better in this species, and even using paraffin paper to wrap around the joining of scion and rootstock. Okay, so right now, at the moment, new grafts of the low nicotine producing lines, the A622, PMT, and double mutant, are growing on wild type rootstocks. So again, I would like to talk with you about the conclusions. The first one is to continue working with the grafting and micrografting, evaluating these some of the protocols in the literature, and also to wait more days until have more material, either more seedlings for the micrograftings and more plants for the grafting assays. Also for the micrograftings, it is suggested to wait not 3 days, but between 7 and 14 to evaluate micrograft success, and also to create control grafts between the same genotypes, either wild type scion, wild type rootstock, A622 scion with the same rootstock, and go on. So, thank you very much.